Imagine you have two patients with the exact same aggressive cancer.
Right. A really tough diagnosis.
Exactly. And you give them both the exact same cutting-edge immunotherapy. One patient's immune system wakes up and just completely obliterates the tumor, but the other patient, zero response.
It's basically the biggest mystery in modern oncology right there.
It really is. And for decades, doctors have just stared at this problem. But a new 2025 study has finally found an answer, and it all comes down to, like, a microscopic turf war over makeshift military bases built inside the tumors themselves.
It sounds like sci-fi, but it is real biology.
Yeah, and that is the mission of our deep dive today. We're looking at a groundbreaking paper from the journal Cell Death and Disease. This study zeroes in on a really stubborn group of cancers called soft tissue sarcomas, or STSs.
And what these researchers uncovered, honestly, it fundamentally changes how we think about the immune system's ability to fight back.
So to start, I feel like we need to understand just how complex these soft tissue sarcomas actually are, right?
Right. Oh, absolutely. I mean, these are rare cancers that pop up in the connective tissues of the body. So we're talking about your fat, your muscle, blood vessels, deep skin tissues.
So they can kind of appear anywhere.
Exactly. And the World Health Organization actually classifies about 70 different histological and molecular subtypes of STS.
70? Wow.
Yeah, it's a massive biochemical variety. And because of that extreme heterogeneity, treating them is just notoriously difficult. Like, a standard one-size-fits-all chemotherapy approach usually fails.
Because the tumors are just built differently on a cellular level, right?
Spot on. They're too diverse for a blunt instrument like standard chemo.
Which, makes sense why there's been so much hope placed on immune checkpoint inhibitors, like PD-1 inhibitors.
Right, because in theory, those drugs don't target the tumor's specific molecular structure.
Right, they target the immune system itself. They basically take the brakes off your T cells so your own body can attack the cancer.
Exactly. But the background data in the study showed wildly inconsistent results for STS patients.
Yeah, looking at the numbers, some subtypes, like undifferentiated pleomorphic sarcomas, saw a 40% objective response rate, which is great.
It is, but then for others, like certain liposarcomas, the response rate was this dismal 6 to 18%.
Right, so the drug is removing the brakes from the immune system, but the car is only moving forward for a fraction of the patients.
That is the central question here. What differentiates the tumor microenvironment of those responders from the non-responders?
And the answer involves something called tertiary lymphoid structures, or TLSs.
Yes. And to understand a TLS, you first have to think about your body's standard lymph nodes, like the ones you can feel in your neck or your armpits when you're sick.
Like the established permanent military bases of the immune system.
Perfect analogy. They are highly organized, permanent structures where your immune system trains its soldiers, specifically the T cells and B cells.
But these tertiary lymphoid structures, they are entirely different, aren't they?
Completely. They are pop-up forward operating bases. The immune system literally builds them on the fly, directly on the front line, usually in response to chronic inflammation or cancer.
What totally fascinated me in the source data was the physical location of these bases.
Oh, the spatial mapping is incredible.
Yeah, the researchers used this technique called immunohistochemistry to physically map the tumors on a cellular level. And they found that nearly 87% of these pop-up bases were built either completely inside the tumor or within just 1 millimeter of the tumor's edge.
Which is just a brilliant strategic move by your immune system. I mean, they aren't building these bases back in some safe zone.
Right, they are dropping them directly on the invasive margin, right, where the tumor is actively trying to breach healthy tissue.
Exactly. It minimizes the distance the immune cells have to travel to reach their targets. But, and this is a huge but, the study makes a really crucial distinction here.
Because simply having a pop-up base isn't enough to save the patient.
Right. There is a massive functional difference between an immature TLS and a mature TLS.
Break that down for us. What does an immature one look like?
Well, an immature structure is essentially just a chaotic gathering of immune cells. Like, the T cells and B cells have arrived at the site, but they aren't organized. They lack infrastructure.
So they're just kind of milling around the construction site?
Pretty much. But a mature TLS, on the other hand, has a highly organized core called a germinal center.
And looking at the biology, they rely heavily on specific cells to mature. You see this really dense packing of B cells, which are marked by a protein called CD20.
Right, the CD20 positive B cells,
Huh?
but they can't do it alone. They are supported by a network of dendritic cells marked by CD23.
And those dendritic cells act as a literal scaffolding, right?
They do. They stretch out and hold the structure together, creating this physical framework. And inside that framework, the B cells undergo an intense maturation process to produce highly targeted antibodies against that specific tumor.
So just to do a quick recap for you listening. Number 1, sarcoma tumors are incredibly complex and hard to treat. Number 2, the immune system tries to fight them by building pop-up bases called TLSs right on the tumor boundary.
And number 3, those bases only work if they mature and form a highly organized germinal center supported by that dendritic cell scaffolding.
Exactly. The researchers found that while having any TLS is better than having none, it is those mature structures that correlate with significantly better survival outcomes for the patient.
But here's where the researchers ran into a serious practical problem. Spotting these tiny temporary structures under a microscope using tissue slices is incredibly prone to sampling error.
Because they're so small.
Exactly. You might slice the tumor tissue just, say, 1 millimeter to the left of a mature TLS, and you completely miss it, which leads to a false negative diagnosis.
So they needed a different way to find them. A genetic footprint, basically, so they could detect these mature bases without having to physically see them on a slide.
Right. So they turned to RNA sequencing. This lets us look at the entire transcriptomic profile of the tumor. And they identified a highly specific 7-gene signature that acts as a blueprint for these germinal center B cells.
Okay, let's list these out because this is the core of the discovery. The 7 genes are CD79A, CD17B, AICDA, TNFR-SF13C, TNFR-SF17, IRF4, and CCR6.
I know that sounds like a bowl of alphabet soup.
It really does. But I was looking at this genetic fingerprint, and one of these genes, AICDA, absolutely blew my mind.
Ah, yes, the mutation engine.
Yeah. It produces an enzyme that intentionally causes mutations in the B cell's own DNA.
Which sounds completely counterintuitive, right? Mutating your own DNA, especially when we're talking about cancer.
It sounds like a terrible idea.
But here is how it acts as a rapid evolution training camp. The process is called somatic hypermutation.
Okay, walk us through that.
So when a B cell recognizes a tumor antigen, think of the antigen as a lock and the B cell's antibody as a key.
Okay, lock and key. Got it.
The fit usually isn't perfect at first. It's like a rusty key. So the AICDA enzyme introduces random, rapid mutations into the specific DNA sequence that codes for that antibody.
Wow.
The B cells then test their newly mutated keys against the lock.
And the ones that fit worse are just discarded, right?
Exactly. But the ones that fit slightly better are kept, and the process repeats. Mutate, test, keep the best.
That is just incredible. And that mechanism has a name, right?
Yes, affinity maturation. By the time the B cell leaves that germinal center, its antibodies bind to the tumor with incredible precision and strength.
That is amazing. And the other genes in that signature support this whole operation. Like CD79A and B, they form the structural components of the B cell receptor itself.
Right, the actual hardware the cell uses to detect things. And then you have IRF4, which acts as a transcription factor.
Like the starting gun that initiates the formation of the germinal center in the first place.
Spot on. Without IRF4, the whole process never even kicks off.
And I noticed CCR6 is in there too. I was reading that this gene controls the homing mechanism, basically giving the B cells GPS coordinates so they know exactly where to go within the base.
Yes, it is all highly orchestrated. And when the researchers analyzed the patient's RNA data, the results were definitive. Patients with a high expression of this specific 7-gene signature had significantly longer relapse-free survival.
Meaning their tumors were actively building mature B cell bases, and those bases were winning.
Exactly. The beauty of this molecular signature is that it captures the functional activity of the immune response, not just the physical presence of some random cells.
But they had to make sure this wasn't just a fluke, right? Like an anomaly isolated to their small 31-patient cohort.
Oh, definitely. So they validated the signature against massive external data sets. They applied it to the TCGA-SARC dataset, which holds RNA sequencing data for 259 sarcoma patients.
Okay, hold up.
Perfectly. The predictive power was incredibly strong. They even crossed tumor types, testing it against public datasets for head and neck squamous cell carcinomas and liver cancers.
So it's not just sarcomas. Across the board, this 7-gene footprint reliably identified mature TLSs and correlated with a better prognosis.
Exactly. So we have this genetic fingerprint. We know these mature bases are vital for survival. But here is the next big question.
Right. The B cells don't just magically arrange themselves into a perfect germinal center. Someone has to lay the foundation and direct the construction.
Precisely. And when the researchers combed through the genetic data of the tumors with high B-cell signatures, they found a massive upregulation of genes specific to a cell called T helper 17, or Th17.
Now, looking at immunological history, Th17 cells have a pretty complicated reputation, right?
To put it mildly, yeah. They're frequently associated with driving chronic harmful inflammation in autoimmune diseases like psoriasis or rheumatoid arthritis.
So I was definitely surprised to see them framed as the good guys in this study.
You and every other immunologist.
Yeah.
The role of Th17 cells in oncology is actually one of the most hotly debated topics in the field right now.
Really? Why is that?
Well, in certain microenvironments, their highly inflammatory nature can actually promote angiogenesis, which is new blood vessel growth that ends up feeding the tumor.
Oh, so they can accidentally help the cancer.
Exactly. But biology is entirely dependent on context. In soft tissue sarcomas, the data clearly shows these Th17 cells taking on a highly specialized beneficial role as lymphoid tissue inducers.
Okay, I love that term. They are essentially the architectural foreman of the tumor microenvironment.
That's a great way to picture it. When chronic inflammation occurs at the tumor site, these Th17 cells migrate in and begin secreting specific cytokines, predominantly one called interleukin-17 or IL-17.
And cytokines are basically chemical messenger signals, right?
Right, and this IL-17 acts as a signal to the local structural cells, the fibroblasts and endothelial cells, essentially commanding them to prepare for base construction.
And how do those local cells respond to the foreman's orders?
Well, in response to the IL-17, those structural cells start pumping out chemokines.
Which are different from cytokines.
Good question. Chemokines are a specific type of cytokine that act like a homing beacon. They pull B cells and dendritic cells out of the circulating bloodstream and draw them directly into the tumor bed.
Like a magnet pulling in the construction materials and organizing them into those highly structured germinal centers.
Exactly. And the researchers actually confirmed this spatially. Using a special stain for IL-17A, they went back to the microscope and physically located these Th17-like cells.
And where were they?
Predominantly inside the fully mature intratumoral TLSs. They are standing right in the middle of the bases they helped construct.
Wow, so the foremen actually stay on site. And naturally, when they applied this to the 259-patient data set, a high Th17 genetic signature was a strong marker for a highly favorable overall survival rate.
And this is where the plot thickens. This introduces a massive biological conflict.
Right, because if Th17 cells are actively trying to build these bases, why do so many patients only have immature, disorganized structures? What is causing the construction to fail?
Exactly. To find out, the researchers inverted their search. They analyzed the transcriptomes of tumors with a low germinal center B-cell signature.
The patients with poor survival outcomes.
Right. And that is where they found the saboteurs. In those failing microenvironments, there was a massive spike in genes associated with M2-like macrophages.
Okay, this is where it gets really interesting. Let's look at how these macrophages operate, because they have this crazy duality to them.
They really do. So in a healthy immune response, a macrophage, which literally translates to big eater, roams around engulfing dead cells and pathogens.
Like a microscopic Pac-Man.
Basically, yeah. But they exhibit incredible plasticity. When you get a cut, an M1 macrophage causes inflammation to fight off infection.
So M1 is the fighter.
Right. But once the infection is clear, it switches to an M2 state. The M2 state is all about wound healing. It dampens the immune response, promotes tissue repair, and builds new blood vessels.
And tumors are incredibly manipulative, aren't they? They secrete chemicals that hijack the surrounding macrophages, forcing them into that M2-like state.
Exactly. The M2 macrophage is tricked into perceiving the growing tumor as an unhealing wound.
That is so insidious.
It is. It begins aggressively protecting the cancer, suppressing any T cells that try to attack, and feeding the tumor with those new blood vessels.
And the researchers tracked these corrupted M2-like macrophages using a specific protein marker, right? CD163.
Yes, CD163. And when they mapped the location of these CD163 positive cells, the spatial evidence revealed a literal cellular turf war.
Yeah, the data show that where you find high concentrations of M2 macrophages, you find almost no B cells. these CD163 positive cells were found swarming the immature TLSs.
Right, if the Th17 cells are the foreman trying to build the base, these M2 macrophages are like corrupt city inspectors.
Oh, I love that. They show up to the construction site and start issuing fake stop work orders.
Precisely. And in biological terms, those stop work orders are specific molecules like SPP1 and MIF.
Let's talk about SPP1 because that gene seems particularly nasty.
It is. Secreted phosphoprotein 1, or SPP1, is heavily utilized by these tumor-associated macrophages to create a localized zone of profound immune suppression.
So it's basically throwing a wet blanket over the whole area.
But worse than that, it directly interacts with the T cells and B cells arriving at the site, triggering a process called cellular exhaustion.
Cellular exhaustion. Break that down for us. What does that actually mean for the immune cell?
So when immune cells are constantly exposed to both the tumor's antigens and suppressive signals like SPP1, their receptors get overwhelmed. They essentially get burnt out.
Like they've been working too many double shifts.
Exactly. They upregulate their own inhibitory receptors and they lose their effector function. They literally go to sleep right on the battlefield.
Man. So by drowning the microenvironment in SPP1, the M2 macrophages actively halt the maturation process of the TLS.
Yes. They prevent the B cells from organizing, they prevent the dendritic cells from forming that structural scaffolding, and they stop the germinal center from ever developing.
The construction is just stopped dead in its tracks.
Yep. And the victor of this microscopic tug of war between the Th17 cells and the M2 macrophages completely determines the patient's prognosis.
Okay, so let's do another quick recap here to tie all this biology together for you listening. We have this pop-up immune base trying to mature. We have Th17 cells acting as foremen trying to build it. And we have corrupted M2 macrophages acting as saboteurs trying to shut it down.
Exactly. It's an active war zone.
So looking at this integrated signature reminds me of like military logistics or even an ecological predator-prey model.
How so?
Well, you can't just look at the supply line. You can't just look at the enemy troops. You have to calculate the balance of power. And the researchers realized this, right? They created a super predictor.
They did. They realized that looking at just one element wasn't enough. So they combined the genetic data for all three variables, the B cells, the Th17 cells, and M2 macrophages.
And they identified what they call the ultimate predictive trifecta.
Right. Patients who possessed high germinal center B cell genes, high Th17 genes, and crucially, a low fraction of M2 macrophages had vastly superior overall survival outcomes compared to any other combination.
That makes total sense. You need the materials, you need the foremen, and you need the saboteurs to be out of the picture.
Exactly. And this integrated model drastically outperformed using just the B cell signature alone or just looking at macrophage density alone.
It just empirically proves that predicting cancer survival requires analyzing the entire ecosystem of the tumor microenvironment.
It's all connected.
But, you know, the clinical relevance of this deep dive isn't just about giving a patient a more accurate prognosis, is it? It's about actionable intelligence.
Oh, 100%. If we understand the exact mechanics of this turf war, we can intervene.
And that is where the future of oncology is heading. If we know that M2 macrophages, specifically the ones utilizing SPP1, are the exact saboteurs halting TLS maturation, we can design therapies to neutralize them.
Yes. And there are currently drugs in development aimed at depleting CD163 positive macrophages or blocking the SPP1 pathway entirely.
So you remove the corrupt inspectors from the site, allowing the natural immune response to just resume building.
Exactly. Or you can tackle it from the other side by reinforcing the architects.
Okay, how would we do that?
Well, we could theoretically design treatments that synthetically boost the Th17 cells or flood the tumor microenvironment with synthetic IL-17 signals.
So you bypass the cells and just provide the chemical orders directly. You force the local structural cells to start producing chemokines, mechanically inducing the tumor to build these mature forward operating bases.
Right. By pharmacologically forcing a tumor to construct mature TLSs, you are fundamentally remodeling the battlefield.
You take a cold tumor one that completely ignores standard immunotherapy because it has no immune infiltration, and you turn it into a hot tumor.
Precisely. Once those mature germinal centers are established, the tumor becomes highly vulnerable to standard treatments like the PD-1 checkpoint inhibitors we discussed earlier.
You set the stage before you deploy the drugs. We aren't just trying to poison the tumor directly anymore. We are manipulating the biological infrastructure of the cancer itself.
It is a massive paradigm shift. The mystery of why some soft tissue sarcomas respond to treatment while others don't is no longer a black box.
It really just comes down to whether the immune system can successfully build and mature tertiary lymphoid structures right on the tumor's invasive margin.
And that success is entirely dependent on whether the Th17 cells can outpace the suppressive, exhausting tactics of those M2 macrophages.
This research provides such a clear genetic blueprint to measure that battle in real time. It offers a much clearer path forward for patients dealing with these highly heterogeneous cancers.
It does, but it also leaves us with a final, very provocative concept to consider.
What's the next leap?
Well, think about it. If our immune system possesses the inherent capability to build these highly efficient, localized anti-tumor factories on demand,
Utilizing specific signaling pathways like IL-17 and organizing through B-cell affinity maturation.
yeah, right. Could we eventually develop a TLS vaccine?
Wait, a vaccine?
Yeah, a treatment that doesn't just broadly stimulate immune cells, but actually provides the exact architectural blueprints, instructing the body to construct these mature germinal centers inside any solid tumor, regardless of the cancer type.
Wow. A vaccine that builds custom immune fortresses right inside the cancer.
Exactly. It's the ultimate inside job.
That is a future worth fighting for. And that wraps up our look at this incredible study. Thank you for joining us on this deep dive. To everyone listening, keep questioning the world around you, keep looking for those hidden biological architectures, and we'll see you next time.